Have you ever wondered about the cure for diseases that deal with genes such as cancer, hemophilia, and cystic fibrosis? We will be interviewing Dr. Fisher on how gene therapy may be a solution to something like prostate cancer in the near future. For that, let's understand what gene therapy is. A genetic mutation occurs when the sequence of a gene has been altered. If a mutated gene causes a necessary protein to be defective or missing, gene therapy can replace the mutated gene that causes disease with a healthy copy of the gene. Gene therapy uses a vector or a vehicle that is best suited to deliver genetic material to the specific target cells so that the DNA enters the affected cells. This allows the new genes to be integrated into the nucleus of the host cell. Finally, the gene must be activated or transcribed to make the functional protein needed. There is no perfect vector to treat every disorder, so the vector needs to be customized for the specific disorder. Viruses have the ability to get their own genetic material inside human cells and consist of genetic material surrounded by a protective coat made of proteins, so they are good vectors. To trick the virus, scientists retain the outer viral coat but modify the inner genetic material. They remove the harmful genes and replace them with therapeutic ones. Now the virus is no longer harmful to the cell it infects. However, it retains its capability to transfer its genetic material to the cells. Some types of viruses, such as retroviruses, integrate their genetic material into a chromosome in the human cell. Other viruses, such as adenoviruses, introduce their DNA into the nucleus of the cell, not in chromosomes. The vector can be injected or be given by IV into the patient by a process known as in vivo. Or the gene can be delivered to cells that have been removed from the body and then the cells are put back into the patient by ex vivo gene therapy. Hello and welcome to Science Talk with Kavya and ABC. Today we're going to talk about gene therapy. Gene therapy opens up the treatment of so many diseases that don't have a cure at the moment. To hear about the successes of this method, I have invited Dr. Paul B. Fisher, who has recently developed a drug to eradicate prostate cancer, to come talk about his discovery. Hi Dr. Fisher, thank you so much for coming.
No problem, happy to be here.
So, before I ask you about your amazing accomplishment, what has gene therapy already been able to cure?
So the first patient to be treated with gene therapy was a 4-year-old girl treated at the NIH Clinical Center back in 1990. She had congenital disease called adenosine deaminase deficiency, called ADA, which severely affects one's ability to fight infections. For the therapy, her white blood cells were taken from her and inserted with the correct gene for making ADA, and then re-injected into her by Dr. W. French Anderson. He's from the National Heart Institute. After safety tests and figuring out what vector to use, the treatment was a success. And since then, experiments for hemophilia, cystic fibrosis, and even certain cancers took place. So we're slowly finding a way to treat these.
Wow, I never knew that this treatment has been going on, the development for this treatment has been going on for so many years. So that's where it all started. Now, tell me about your experimental breakthrough for with eradicating prostate cancer.
We used a cancer terminator virus called CTV, which is a modified adenovirus that replicates exclusively in cancer cells, delivering the cancer-specific toxic cytokine gene directly to the tumor. We added an experimental drug, BI97B6, that targets the specific gene type BCL2 that protects cancer cells from dying.
So ultimately, what was the drug able to do?
This allowed the cancer cells to be killed by the CTV, and it also helped defend against resistance to the viral gene therapy. Ultimately, the prostate cancer cell died, while the healthy prostate epithelial cells remain unaffected. As the CTV replicates, the gene that kills the cancer cells is secreted and prevents tumor growth.
So how do you ensure that the vector reach the targeted area?
We use a technique which is called ultrasound targeted microbubble destruction, UTMD. This uses microscopic gas-filled bubbles that can be paired with viral therapies, therapeutic genes and proteins, and imaging agents. These bubbles are released in the targeted area via ultrasound, and with appropriate modification of therapeutic virus can be imaged and tracked in real time of the delivery of the CTV to the tumor.
Wow, that is truly amazing. It's really cool stuff. So at the moment, is gene therapy being able to be used extensively?
Well, at the moment, only clinical trials are going on, and hopefully it will advance further. The problem is that there is definitely room for things to go wrong, and we need to be careful. Viruses can trigger one's immune system as they're seen as intruders, which will attack the viruses. As a result, the patient will fall ill. And there is always a possibility of the vector attacking the wrong cell, which could cause other illnesses. So we have to proceed with a lot of caution and care to make sure that gene therapy is safe for our patients.
Okay. Thank you so much for coming today. This was a really good talk, and we hope for this treatment to flourish and move forward. And thank you for tuning in into ABC with Science Talk.
Thank you.